Alright guys, welcome back to Underground Science, and in this video we'll be going over the interaction between a helper T cell and a CD4 positive cell. Alright, so just to get things started, another word or another name for a CD4 positive cell is also just a helper T cell. Alright, so a helper T cell is also known as a CD4 positive cell, and we'll see why it's called a CD4 positive cell in just a bit. So remember in our previous video when we talked about antigen presentation, we talked about the fact that B lymphocytes are one of the many professional antigen presenting cells. So let's go ahead and draw us a B lymphocyte here. Alright, so let's go and say that this is our B lymphocyte, alright, with its nucleus, and let's go and draw some more color, right? So let's say this is our B lymphocyte, and we have our MHC2 complexes, right, present on the cell membrane, right, because this is how it's going to present its antigen. And I don't think I mentioned this in the antigen presentation video, but the full form of MHC2 is good to know, right? So let's go and write that down way down here, right? So the full form of MHC2 complex is the major histocompatibility major histocompatibility complex, all right, type 2. So that's what MHC2 stands for. So that's why we'll be just saying MHC2 instead of saying major histocompatibility complex type 2 every time, all right? So these are our, sorry about that, these are our MHC2 complexes on the surface of our B lymphocyte. All right, so let's go and label these out so we know what's going on. All right, and so let's say, right, so let's actually go and draw another one right here. It's going to be our main one that we're going to be talking about, right? So let's go and draw this one out here. It's another MHC2 complex, and let's say we beforehand, right, this B lymphocyte came across. Oh yeah, we have to draw our membrane-bound antibodies also. So let's go and draw our membrane-bound antibodies with our heavy and light chains, or at least try to show that, right? So let's just draw two for now. So we have membrane-bound antibodies, right? And so let's say we have some type of antigen that's already being presented on our major or our MHC2 complex. So let's say this is an antigen, right? So this is our antigen. So let's say beforehand, maybe our B lymphocyte, right, took in a pathogen, broke it up, then presented it on its surface. All right, so we have a pathogen protein right here. We have an antigen protein right here. That's part of the broken up pieces of the pathogen, whatever virus, bacteria, fungi, or whatever it was. All right, and so remember we said that when we talked about B lymphocytes, we said that it can bind to specific pathogens on the antibodies, right? And start proliferating into memory B cells and plasma B cells. But however, right, that was just a basic overview. We said we needed another very important thing, which was our T cells, especially our helper T cells. So what can happen is that along with these membrane-bound antibodies, this part is really important too, because we need a helper T cell in order for this B lymphocyte to be active. So we can start dividing. We need a helper, and that helper is our helper T cell. All right, so our helper T cell comes along, right? Our helper T cell comes along, so let's go and draw this out, I guess in this orange color. So our helper T cell comes along. Let's say this is our helper T cell. All right, and I drew that a little too far away. Sorry about that. Let's go and draw just a tad bit closer. So it's our helper T cells right here. And our helper T cell is going to have a T cell receptor on it. Okay, so it's going to have a T cell receptor on it. We can talk about alpha beta T cells and gamma delta T cells later, but this, just to know right now, right, is that we're going to have a T cell receptor on here. So let's see what color is just doing gray. So we're going to have a T cell receptor that will actually has to match to the antigen in order to bind to it, right? Just not one random T cell is going to come and bind. Whatever T cell that has the matching antigen binding spot on its T cell receptor is going to bind. So this is our helper T cell, all right? That's coming along to help. And so once it binds, this B lymphocyte is still not active because we need another part. And this is why helper T cells are called CD4 positive cells, because these helper T cells will also have another glycoprotein called a CD4 protein that's actually going to extend out and latch on to the major histocompatibility complex type 2 protein on the B lymphocyte. So we can go and draw it like this, right? And now we can say that it's once it latches on, so this is our CD4 protein on our helper T cell. So once our CD4 protein binds onto the MHC2 complex, and then once our T cell receptor, so let's go and label all these parts here, there's so many parts to label, right? So this is our T cell receptor. So once our T cell receptor binds to the antigen spot, and once our CD4 protein of our helper T cell binds to the MHC2 of the B lymphocyte, then our B lymphocyte can become active, right, after our helper T cell will actually release some lymphokines, right, or other types of cytokines also. So we're just going to say it will release some lymphokines and cytokines. So pretty much it's this. So after our CD4 protein binds to the MHC2, our T cell receptor also has to be attached to the antigen's binding spot of our MHC2, right? And so after that happens, our T cell will actually release, our helper T cell will start releasing certain types of lymphokines and cytokines. So pretty much inflammatory chemicals, all right? So these are going to be our lymphokines, and cytokines. Specific types of cytokines and lymphokines will be released from the helper T cell once our CD4 protein binds to the MHC2 complex and once our T cell receptor binds to the antigen that's present on top of the MHC2 complex on the B lymphocyte. Helper T cell will release some lymphokines and cytokines, which will then travel over to the B cells, right, and can bind onto the B cells and actually induce proliferation of the B cells or the B lymphocytes. So once this happens, we can go and bring this down here, right? We can go and bring this down here, and once this happens, our B cells can actually start proliferating into memory B cells, and into B effector cells, or also called plasma B cells. All right. And so what this is going to be, right? So we're going to have a bunch of memory B cells here with the same, so we have to remember, so sorry about that. We're going to have a bunch of memory B cells there with the same variable portions of our memory-bound antibodies also. All right, so we can have the same membrane-bound antibodies here drawn. It's not going to be a different binding spot for our membrane-bound antibodies because remember, we're trying to make more B cells for that specific pathogen that caused us to divide. So it wouldn't make sense to have different membrane-bound antibodies because we're trying to attack more of this specific virus or bacteria, whatever pathogen that we have presented on its cell. All right, so we can, and also note that there can also be the same pathogen as a whole, right, even bound to this membrane-bound antibody also, right? So we have a lot going on here, but eventually we're going to have proliferation and we're going to have our major histocompatibility complexes, right, on our memory B cells, and these memory B cells are then going to be kept for later so that we can use it for later once we have a reintroduction of the same pathogen that originally induced proliferation. All right. so we can attack the same pathogen later, once it comes later, and we can get rid of it much faster and take care of it much faster. All right, and on the other hand, our plasma B cells are pretty much B cells that will just start kicking out a bunch of antibodies. All right, so a bunch of antibodies for that specific antigen that originally binded onto the membrane-bound antibody. All right, so it's going to start kicking out a bunch of antibodies. So it's going to draw a bunch of antibodies, right, that our plasma cells will start kicking out. And from our last video, we know it's about 1,500 to 2,500 antibodies per second. All right, so we're going to have a bunch of free-floating antibodies that start getting kicked out and start getting produced by the plasma B cells that will go ahead and find the same exact antigen, right, or pathogen that was originally bound to our B lymphocyte, and it'll go and perform opsonization and attract other white blood cells to come and phagocytose those pathogens. All right, so that's pretty cool. We make memory B cells and plasma B cells after activation through the help of our CD4 positive cells, or our helper T cells, and we also will see that not only do the lymphokines and cytokines activate the B cell to proliferate, but what happens is that our B cell, once it becomes active, can also release some cytokines in turn. So it can also release some cytokines. So our B cells, once it's active, can also release some cytokines, and these specific types of cytokines can actually go over to the helper T cell and actually cause the helper T cell to start proliferating also into memory helper T cells and effector helper T cells. So you know what, we'll pick this up in the next video. All right, so see you guys there.